This here. Okay, welcome everybody to Open Source Antibiotics Series 1. The Mur Ligases is on the 10th of May, 2022. Thanks very much to Chris for standing in for me last time. Looked like it went well from the recording. And I think we're going to have some more data be presented on some of the molecules that Adrian's been looking at, which would be great. I did send him a link to the meeting. It was interesting to hear about last time about the SSGCID and their potential existence in the future. I guess we're not quite yet sure. Jan, do you have any more information about the future for the group in Seattle? You hearing me? Can you hear me, Jan?
Sorry, I had headset issues.
Okay, no worries.
And for some reason, the meeting shows up as a Zoom meet, as a Teams meeting on my calendar. So it tries to start Teams.
Yeah, Outlook, I think it guesses that it's Teams, even though I'm pretty sure we didn't set it up like that.
Yeah.
You just have to ignore Outlook. It's mistaken. I just wondered if you had any news about, because I heard on the last meeting that you guys won't be able to necessarily contribute much after about September the 1st. I just wondered if you had any news about SSGCID and their continued funding and existence.
I don't know about the continuation of SSGCID. That's something Bart would be able to answer, because it doesn't really impact us at all anymore. We're still good till, I think it's the end of August.
All right, yes.
So whatever we can do till then is fine, but after that,
great.
I won't be paid for it anymore.
Okay. Okay. All right, duly noted. So yeah, I mean, well, hopefully we can keep on interacting with you, but we understand then that it's not budgeted.
Yeah.
Got it. All right. While we're waiting for others to join, why don't we just run through Kato's very nice summary, which she just shared. I can share it here if that helps. So Kato has been working on her project in the lab and is coming to the end and is going through the nerve-wracking experience of writing up a thesis. And as opposed to a nice summary here of some of the work that she's been working on in the interim. Kato, did you want to just walk us through this and tell us what we're looking at?
Yes, I'll quickly walk through it. I think I created some compounds in the first term, like until December or something, which I call elaboration round 2A. And I think I spent 2 weeks on thinking about how to design them based on the SAR analysis of Dana's elaborated compounds. And we decided to synthesize compounds based on the piperidine core, as I think 4 out of the 6 dual inhibitors have that same core. So we thought it was a good starting point. So we focused on that. So all compounds have the same core. These compounds, I took them to Oxford and we tried to do some crystallography with them. We only managed to do soaking, and I think the results are not, I think they're processed by Laura, but I don't think she saw anything. So that's a bit sad, but yeah. Then in the meantime, I think Becca did her dose response curves on the dual inhibitors. And out of that, we managed to see that the compound was a 759, I think, of elaboration round 1, had a good IC50 value of less than 40 micromolar. I think it was 21 point something. So that looked promising, and then we based another round of compounds on that compound, and that's what I call elaboration round 2B. Didn't manage to purify and characterize them all, so I think Ed will continue on doing that, and after that is finished, I think we can send the whole batch for more crystallography, the enzymatic assay, and SPR. So that's what I did in the last few months.
So the green ones are ready to go. The orange ones are needing a column, and the gray are EDs.
Yes, that's correct. Some are already purified ones, but there are some more impurities that we have to get rid of. The last two orange ones need the first purification as well. So time-wise, I don't really know how long that takes. Ed works a little bit faster than me, so.
All right, Ed, anything to add that? You're on the case for these molecules?
Yeah, they've all been made. They're just at various stages of purification. So hopefully in a week or so, we'll get them done.
Okay. And then the question is what we do with them, right? So, I mean, there are three options here. One is the soaking, one is the SPR, one is the enzymatic inhibition. Is there a reason not to do any of those three?
Happy to have them to put them through enzymes.
Great. Yeah, I mean, that would make a lot of sense because that's what we're basing this on, right? Is Becker's original experiments. So that makes a lot of sense. SPR, I guess, doesn't use very much protein. Is it fine to also try and schedule them for analysis in that if that's not going to be too time consuming?
We can do SPR, but I would do with more compounds because there's a problem with a shortage of sensors. So we need to be smart of how many sensors we use. So when we get the compounds from the competition, I can test many other compounds as well. Okay. So if we want to test these ones on SPR, I can do that as well.
Okay, perfect. We can combine them with more.
Yeah.
And then I guess, you know, the issue with this always was that we had the inhibition data from Becker's experiments, but we didn't have any crystallography with these compounds. Of course, that's what we're aiming for. So I guess, I mean, does it make any sense to try and do crystallography with these, given that we haven't had any luck with the simpler compounds yet?
Yeah, we can. I don't know how much related are these to Kato's compounds. He already tried some soaking on Mur D and Mur E, but I couldn't see any compounds bound on the data that was collected.
Right. So I think I remember from the last, just from looking at the recording earlier, that there was some discussion about including in those trials UMA, ADP, things like that. I guess I'm wondering do you feel as though you want to try and optimize the soak before you look at these next compounds? Is that the order of events?
We already tried with some compounds from Kato. Well, she did it when she was in Diamond with Lizbe. And I was I'm also trying to optimize that condition to do soaking on the more compressed conformation, but I don't have more updates on that because it's not optimizing as well. So
I think the bottom line is we need some more improvements in the crystallography setups.
Yeah. So
and probably the best we can do is do the biochemistry and focus on which series seem to give the most potency. And then if we had a simple crystal soaking system, then yes, we just put them all in, but we haven't at the moment.
Okay.
Yeah, right now there is not a new system that we can try them on. So yeah. But if we have a few compounds that we want to follow up on crystallography because they're looking really good, we can always do co-crystallization.
Okay. So initially, enzymatic is less of a problem than combined with more compounds for the SPR. And then if the crystallography assays are looking better, then we can try the compounds then too. So we should be shipping you several MIGs of each of these, basically.
Cool.
Yeah. Good.
All right. Great.
What are the affinities of these compounds?
We
missed that. Is it? Yeah, I think that is crucial to be successful in crystallography, not to just send many compounds to crystallography, but only send the compounds that show in biochemical assays they are really good binders. Because a lot of time wasted by Laura if the compounds just aren't good enough.
Yeah, so the first set here that's on the screen were the ones that gave some inhibition. But we didn't have any measurements for binding affinity from SPR, for example. So I think that, yeah, you're right in the sense that it would make sense to get that first.
Yeah. I think we should shoot for something. Yeah, go ahead.
I'm going to say that you have to remember though that these are fragments. They're not going to be high affinity ligands at the moment.
Yeah.
And you can, we do fragment screen in crystals to find fragment start points in crystals. I mean, that's definitely doable.
Well, these came from a fragment screen originally, so.
Yeah.
On crystals, yeah.
Yeah. But I think, just prioritizing the most interesting ones is probably the best way to go anyway.
Yeah.
Okay.
Are these well soluble compounds? When you see a lot of heteroatoms.
Most of them have got basic amines in them, so you would imagine they'd be soluble.
Yeah. Because for fragments, we typically screen at 50 millimolar of compound. That requires that they are soluble in DMSO at something like 200 millimolar.
Wait, Kato, didn't we put the log P less than 3 rule for these?
Yeah, the log P, except for one compound, I think the C log P is below 3. And they're quite, when I try to dissolve them in DCM or methanol, it looks fine. I don't, DMSO, I think the ones I took to Oxford, they all dissolved in DMSO. So I think that's actually fine.
Okay.
Yeah, we tend to put that on there now when we're doing these high concentration assays.
Okay.
Okay. Thanks, Kato. Hopefully we'll maybe see you at the next meeting, but it depends how your exams and stuff go. But yeah, Kato's busy writing and submitting and doing exams and stuff. So she may not see a lot of her on the website in the next few weeks. But hopefully we'll get some nice data that you can include in the thesis. All right, and just while we're on the same page, Yuhang, did you want to just quickly update? I think we can leave all the stuff to do with the shopping list compounds, which we are looking at, but I guess just a 1-minute update on where you are with the crucial synthesis of the amine compound. This, but without the Bach group.
Yeah, sure. For the first AZ595 synthesis, I think I've updated last month. Yeah. So I used that optimum condition, applied that condition to the next reaction with the Bach on the starting material. But that reaction turns out to be too harsh. So all my products and starting materials deteriorated during the process. So I started to use the LCMS monitoring every 5 minutes just to carefully handle this reaction. And eventually the condition should be limited to 30 minutes on a 150 Celsius degree microwave. So everything else remains the same as the AZ595 reaction. So, but just to limit the time to 30 minutes, that's it. And the separation is ongoing, need for characterization, and this is a new compound. And about the second bullet point, I think this is something that we need help from Yuhang because I think Peter didn't finish it. I haven't got the result afterwards. I think.
Sorry, can you? I've forgotten in all that's going on. I've forgotten what the aim here is. So the overlay of these two things. What's the purpose of doing this?
Oh, the purpose of doing this is because we want to see how this WH92 product was overlaying with the AZ595 to see if the key interactions are the same. And yeah, so just want to see how the interactions are going with the residues. Are these AZ series of compounds were behaving in a similar way? And also, this compound has a positive control. So that's why we're doing this.
So we're just checking it's doing the same thing. Okay.
Yeah.
All right. So if you can pursue that offline to try and complete that analysis, that would be great.
We haven't got any data. We haven't got any data from Peter or Young. Previously,
Yeah,
I only had a 5-minute. Sorry.
Yeah, please go ahead.
Previously, we only had a 5-minute video Zoom meeting with Peter, and he only briefly showed me the position of this compound in the pocket. Then I haven't got any other data from since then.
Yeah,
I think it's last year.
Okay, I'll follow up.
Okay, right. Thanks.
Yeah.
Thanks. Great.
But,
Okay.
I thought they were already on the PDB, right?
They are.
Yeah.
We can download the data from it. Yeah.
Could you send me your email?
Yeah.
I can.
Sure.
Yeah.
This is Joe. I'll work with Yuhang on this and help him out. I mean, this stuff's already it's all there. I'm not quite sure what the issue is, but I'll follow up and get this sorted out. It's just a trivial thing.
Yeah, I think it's one of those small things that because it keeps getting passed from meeting to meeting, it grows in size. And actually, I think it's quite a small thing to do.
Yeah. Okay, that shouldn't be a problem at all.
Thanks,
Things
Joe.
on action trackers get bigger as time goes by. It's weird. All right. So I guess there are some other key things from last time. I mean, we've covered some of the chemistry here, and we've covered some of the stuff to do with what we're going to do with these other compounds here. There is still an action on me to follow up with the Atomwise guys about the nature of their compounds and where they've bound. They're waiting on us to provide them with a description of exactly what happened and where the compounds are and all the data. We've given them all the links, but they still wanted a description of exactly what we're talking about. It appeared from our call that the predictions of the compounds was slightly fortuitous in the sense that they had predicted compounds to bind and they're binding better because of the change in structure of the protein when the binding took place. But we need to provide them with accurate information so that they can give us their final analysis and potentially suggest some other compounds. So that's stuck with me at the moment, I'm afraid, and I haven't seen that through. And then, so before we get off the subject of the crystallography, Laura, I just wanted to give you a chance to say anything you wanted to about anything more you may have done, or Lizbe about the assays to try and get structures crystallized with small molecules bound, so the soaking experiments. Are there any updates you need to provide on that, or is it work ongoing?
From my end, is work ongoing. I don't have more updates.
Okay. Lizbe, was there anything else? I just want to make sure we don't skip over the important stuff.
No, so all I basically did was, when Kato was here, have her screen a lot of core screens, trying to find if there was other crystal forms or anything that would work better. But past all the data to Laura, it seems that we didn't actually manage to get anything that was better than what you already have. It's a bit sad.
Okay. Okay.
Yeah, it was exactly the same packing on different conditions, and it was packing in the same way for every single one. Yeah.
Okay. Are you able to, I mean, is there something useful to share there about what was tried and therefore what didn't work? We just want to make sure that we're also reporting the negatives if you have anything.
I can upload a little summary of it.
It would be handy. I noticed there was one last time from Kato, and it would just be nice to have something. So, you know, in a few months' time, we don't forget.
Yeah. I can upload.
So Laura, so This is Joe. Have you been trying any systems with UMA included?
Yes, but they were very small, so I'm trying to grow them now.
I'm sorry. So you just got small crystals.
Yeah, so only a small crystal. So I'm trying to grow them now and making them have them more often.
Okay.
So we can try to soak in them. Yeah.
Okay. Okay. Thank you.
No problem.
And I seem to remember there was some action, I think on Laura again, you seem to be getting them all today, about shipping some UMA to SSGCID in Seattle.
Yeah, we have to pause on that because we didn't have enough at the moment, but we do have now. However, Julie's away. Yeah. So I will do it as soon as he's back.
Is it a commercially available material? I wasn't clear about it.
No, we will send it. Yeah.
Okay.
Good.
All right.
As I said, there will be a time limit to our involvement.
Yeah.
Yeah.
So it's May already, and you guys are around until the end of August.
Yeah.
Yeah, Laura, how much?
And probably our bosses would like to finish up earlier than that.
Okay.
Because we have spent the time.
How much do we need to send? Can we just go through our freezer cupboards and pull out whatever we have, and rather than waiting to make it up?
Yeah, we can do that. Would it be okay 1 or 2 milligrams? I think we're talking about 2 milligrams.
Yeah,
something like that. Yeah.
Yeah, let's see if we can get that. What day is it? Tuesday. See if we can get it posted this week.
Yeah.
Okay.
Okay.
Okay, I sent the meeting link to Adrian, but he's not.
Oh, he's in the background.
He's in the background.
Yeah, I'm here.
On your screen.
On Laura.
Oh, on Laura's screen.
Laura, yeah.
You're blurred. It's like you're censoring yourself.
Oops. Wait.
It's all right.
Yeah.
Just, enzymologist in the shadows.
There you go. Hi, Adrian.
Hi.
I just watched the recording from last time. Very interesting data you were showing. Did you want to have any updates today?
I'll be generating an update when we've got a more complete data set, if that's okay.
Oh, sure, no problem. So some of those molecules look pretty interesting, I guess, and we're giving nice results. And it sounds like you've been doing a huge amount of work on the enzymatic assay.
Thank you.
All right, great. And I think you were also talking, so you're talking about, I mean, obviously IC50 values of all compounds and whether they were ATP competitive. Is that still an up-to-date aim?
Yes, it is. Yeah.
Okay, great. Just from what you were talking about last time, it looked like you've measured a bunch of compounds. Do you know roughly how far through that set of compounds you are?
How what?
How far through the set you are? So of the total, how much has been evaluated?
Well, of the entire amine set at the moment, incompletion for allogalactii, and something like around, and we've just taken the allogalactii hits and have characterized those with the other ligases at the moment. And then we're going, once we're happy with the performance of the stopped assay, then we're going to basically go and repeat the enamine set on C through to F.
Okay.
I mean, you can give a bit of a spoiler, Adrian, that you know, that we found at least one compound that triply targets. So
that's amazing.
Yeah, we did. Yes.
We never even talked about triple targeting. We just paused at double targeting. Right? You're going to have to go for the quad now.
Well, yeah, we are thinking about trying to lose MRF as well. We're going to do that.
Okay, so from memory, it was the compounds that Joe and Becca designed. It was like 600 compounds.
Yes. Yeah.
Okay, so there's quite a lot to measure there. Okay, great. All right, wonderful. Thanks. Okay, so we can do that in the June meeting. Hopefully, that would be really wonderful. There was a brief discussion last time about compound numbering. And I guess I just wanted to flag that up because we have resolved this to some extent. But let me just talk about this. So just there was some comment about compound numbering and about being systematic, and there was also a comment about wanting to retain provenance and origin of compounds, which we totally get. So we devised the systematic numbering system, which is the one that's shown on the screen here, which is the one that's used in the database of OSA molecules. So which is an integer plus salt form and plus batch number, which is fairly standard. And of course, if you want to talk about a compound, you just use the number. You don't have to use this massive string, but to be systematic about it, there is a number there. But it's always very convenient in projects like this to have another parallel code, which is more bespoke and which can be used to show provenance. In open source malaria, for example, we've always had part of the code as a letter which indicates the city where the molecule came from, just to try and get that idea across. So in the spreadsheet that we have, besides the systematic number which I think we need, it's absolutely possible to have other identifiers, and that should satisfy everyone who wants to make sure that they've stamped their number on a molecule. Obviously, besides the number, the public nature of the work we're doing, so reports and lab notebooks and what have you, also ensures that there's no contribution which is made which is divorced from the person who's made it. I mean, it's absolutely essential that is maintained. But did anybody have anything in mind other than that when we were talking about compound numbering?
No, it's just good to have it, and and make sure it's widely circulated, and none of us in a month's time saying, where's the numbering system? So we've all adopted it.
No, fine. Okay, so I'll put a link for this into the issue. It is, the reason it's maybe not easy to find is because it's in the so-called tech ops wiki, right, which is all the dull stuff about how we do things. So I'll make sure that it's cross-linked to this issue and cross-linked somewhere on our main repository, so it's easy to find. As well as the link to the actual list itself with all the molecules.
And it's a single digit allocation for the city.
It can be anything. No. So the number here is where there's a rule, but your code, your internal code. I mean, we in the lab books we use in UCL, we have codes that are people's initials, and then the reaction number and the attempt number and blah blah blah. If you have a library of molecules, you probably have your own numbering already, like Diamond and Warwick. You have your own codes, and those can just go in to the spreadsheet in a separate column, which is in there already.
Sure. And if you wanted to put an avatar in there, you can put your own favorite avatar.
I just think that's quite important because we all have different systems, but at the same time, we do need to agree on one number.
It's very useful though if you tell people to avoid certain characters which don't necessarily get transferred. So if you stick to alphanumeric things and avoid slashes and things like this, because some systems will just treat them as arithmetic operators and things like this.
It's amazing. I mean, when I was told by the people who know much more about this than I do, like Chris, for example, that the underscore is still useful, and that reminds me of using like Windows 95 and naming files with underscores, so the computer doesn't get confused. But that still is it's unambiguous and useful in a way that apparently a hyphen is not, because there are 3 different kinds of hyphen that you can use, right? And spaces are a no-no. So we used to have, in open source malaria, it used to be OSM. So sometimes there was a space there, and it was like, no, it's a horror, apparently, according to the cheminformaticians. So something with underscores is good, or with no gaps at all is even better. But then sometimes it's more difficult to pass those. But in your local synonyms for molecules in your local codes, yes, as Chris says, if you can avoid spaces and other ambiguous characters.
Great.
It's too late to change it for open source malaria. Hyphens are everywhere, unfortunately. So we're just doomed, I think. But we can at least try here. All right, good. those were the main things. Oh yes, there was a question also, a discussion about funding mechanisms, which was very interesting. I'll put up some notes about that when I managed to write up the notes from last time. In last time's notes, a summary of the meeting. The discussion of R21 seems like a sensible idea. I mean, Chris, Dowson, and I need to keep an eye on funding mechanisms over here too. The thing that was mentioned about the NIAID program for making molecules for AMR, yeah, I'll put a link to that. It's this cc4carbcollection.org thing, which I'll put a link to. It does look interesting, and I meant to raise that at a previous meeting, that we should be checking that out as a potential source of funds. And what was the other thing that was mentioned? I forget. Oh yeah, and the CarbX idea. I'd agree with what Joe said last time, that we're not yet in the zone for anything like that with the current level of data. As always, we have been aiming for molecules with better MIC values before we go for funding, but certainly for CarbX, you need something that's much more advanced than we have, I think. I'm giving a talk about open source antibiotics at the Gordon conference on antibacterials in July, at the end of July. So I'll summarize this project and what we're doing. And there are some really interesting people going to that who are from funding agencies, and it's going to be good to talk to people there about possibilities. All right, those were the big things, I think, from last time, and I don't think we need to dwell on anything else because I think everything else is pretty clear. So I just wanted to throw it open to anybody else to talk about anything that we haven't yet mentioned.
I wrote to the Merck open source people, sent in and got a very warm response from them, but I haven't found a proper response saying yes, you can have the compounds. So I'm just going to go back and check through my junk mail. Okay.
Does it look onerous?
Well, the form I filled in was not onerous because I'd written most of those words, and it was just cut and paste from the ELF actually. And yeah, no, and they said yes, we're very enthusiastic about supporting antibiotic discovery, and that was the last email I got. So I need to go back and just make sure I haven't got another one I've lost. Or
is it? I'm sorry if I missed this. Is there some? Is the library unusual and or large? Do we know anything about the composition?
I can't remember what the composition is, but it comes unfettered, IP unfettered, which was the appealing part of it.
Okay, it's also, it mentions infectious disease research. So, I mean, it sounds really promising.
Yeah.
Yeah, I don't think that there was any specific details on the website in terms of the actual contents of the library. I think it's just made up of their, in-house research compounds that they've generated for other programs.
Right.
Yeah, Chris, we've had a similar collaboration, also asked to reach out for this library for kinetoplastid diseases, and when they got that reply email, they attached an MTA to it. So if they forgot to attach the MTA, maybe that's all that you actually need. But yeah, I mean, it sounded like it was a really interesting opportunity in terms of having compounds that are drug-like, but I have no idea what the actual contents of the library looks like.
No, let me go back and go and chase that then.
Right. A quick update on the competition that we're running. We've so Jan and Casper Stein have submitted some compounds that that Ed and I are currently working on about whether we can buy or make. 2 other inputs have come in from 2 new participants. So we need to look at those too. They came in just in the last 24 hours, and we're expecting some more. And we just need to think about how we're going to secure these molecules as quickly as we can. So these are all using so-called generative methods in AI. Well, to the extent that it's possible for the entrants to use those methods, they're using them. And we may get some interesting suggestions. Let's see. We're going to try and get them as quickly as possible so that we can send them to Laura for evaluation, but we'll keep you all posted. We're starting to get the inputs now, which is good. we have reached out in a targeted way to a bunch of companies working in this area, and we're going to try and do the same with academic groups now as well.
Yeah, thanks, Laurie. I've just found the email, but our Warwick mail protection system has managed to delete the attachment. So I read it.
Oh boy!
And there's no attachment. So I'm gonna reply to them. May I use my private email address or something?
Yeah.
Go another route. Yeah, thanks for that.
No worries. Sorry.
That's right.
Okay, good. I have nothing else. So last chance for.
Oh, this is Joe. So I guess I'll pipe up here. So Chris's group has done a fantastic job, the last, whatever it's been, 6 months, a year, getting the biochemical assay in place. So that really seems to be in a great position right now. So I think the obvious struggle, and this is not meant to, anyway, the obvious right now, we need crystallography. We need to get crystal structures of these compounds. I think that's really essential probably for any kind of grant funding. And I'm just wondering if there's worthwhile for kind of a special meeting or brainstorming session to kind of go through what's been done and maybe some thoughts about what really artists. I know, Laura, I know you're working hard on this. This is not meant to be. This is not a trying not to be Monday morning quarterbacking here, but just in terms of, I think this is the nut we have to crack in terms of crystallography, and figuring out, what are the various ways and what resources we have to try to make progress there. Because I think that, at least from my perspective, I don't think we can get at least any NIH funding without probably crystal structures of some of these compounds. So anyway, just the thought of, is it worthwhile having any kind of I'll call it crystallography summit or something where the experts here get together and try to see if, what stones have been left unturned, maybe put it that way.
I think it's a good idea.
That would be great. I mean, fantastic from my perspective. If people are willing to have a subgroup meet and talk about that, that'd be really good.
Yeah, that's good.
I suppose in advance of that meeting, it would be, I mean, again, it's I don't want to put too many tasks on people, but if we could somewhere assemble a very short description of exactly what's been tried so far, that might help us in determining where the gaps might be. If people are willing to put together a few slides that we can have a look at to be clear about that, because we mention in the meetings and we try and take notes and things, but it'd be good just to go through the kind of grid of different proteins and where they come from and what's been attempted. That would be pretty useful.
Actually, one thought just came to mind almost even. So even if, if we do I can never say it, the galacti system, Mur D, granted it's not a gram negative, but I don't know, do you think that's any more robust to potentially try some of the hits against that system? I mean, obviously we want to ideally get something against Pseudomonas or E. coli or Acinetobacter would be the preferred species, but if we had if you had the biochemical data to show that you're hitting several of the gram negatives as well as like the Mur D galacti structure species.
The crystals of Agalactiae take about 2 months to grow.
Okay, I get started. No, sorry. No.
Yeah, but it's not. They're not. So you might get 2 or 3 crystals. So you need to set up about 20 plates to be able to do a screening. It's not a good system.
Okay. All right. Again, those are the we call it the practicalities that you know about and so on. So, and that obviously I don't know. No, I was just trying to say that sometimes if we could get if there's an other Mur Ligase species that was close enough that made sense that you thought you could get better, might be more robust in terms of crystallography. That was just the thought.
Hmm.
Yeah.
Just as an FYI, Joe, when we actually found the Agalactiae system, previous postdoc Dom Bellini must have looked at about 15 others as well. And so that was the best of a bad job.
Okay, okay, good. But again, that's kind of what Matt was talking about. It's kind of just, I guess, as a community, knowing kind of, you know, some of that history in the context of what we're trying and where we wanted, where we might go to try to find success.
Yeah.
Okay, so maybe we could aim for that in between now and the next meeting. So maybe at the end of the month or something. That kind of idea.
Yeah, that's okay.
All right, good. I can send around some times and see if we can meet up at some useful time.
I will be on vacation the next couple of weeks, so I won't be back before the 24th.
24th, right? We'll do it after the 24th. I think you need it.
Yeah.
I'm away next week too, so yeah,
okay.
24th is good.
That's when I'm returning. So
yeah, fine.
25th onwards.
We'll let you unpack your suitcase.
Yeah. Okay.
Monday morning quarterbacking. I have no idea what that means, but it sounds good.
I don't think it is.
Come on,
I don't think it is.
Like, yeah, it would just be like you after, you know, Liverpool beats your ass, you know, in some game. You're saying, oh, why didn't the coach do this, or why didn't that player pass the ball there? And you know, it's basically second guess. It's the version of second guessing American football players, and that's what it's about.
I see. Got it.
I've been on the receiving end of AZ Monday morning quarterbacking for a project that wasn't going very well. I know what it feels like.
All right, great. Thank you for that, Joe. Good point and good idea. All right, if there's nothing else, then we can end at 1:45. Very nice to see everybody. Thank you very much for coming along. I'll put up some notes online, and our next meeting is again the second Tuesday next month. But we'll have our crystallography soak in towards the end of the month.
Sounds good.
All right.
Appropriate name.
Thanks everybody.
All right.
Bye-bye.
Bye then.
Bye-bye.
Bye.
Bye.